protein, primary, secondary, tertiary, and quaternary structures. Proteins and peptides are formed from amino acids connected together via peptide bonds. Peptide bonds usually form between two amino acids, where one of them loses a hydroxyl group and the other loses hydrogen atom. This process leads to the formation of the peptide bond NH3'O. If both peptides and proteins are formed of amino acids connected via peptide bonds, so what are the differences between them? In terms of structures, peptides consist of relatively small number of amino acids, usually less than 50 amino acids. These amino acids are joined together through peptide bonds, which creates a linear chain of amino acids. In contrast, proteins consist of more than 50 amino acids that are connected by peptide bonds. This results in a longer unbranched sequence of amino acids, which can further fold into more complex structures. In terms of function, peptides serve only as regulator of specific proteins' activities, while proteins have many functions, including structural support, enzymatic reactions, transport, hormonal signaling, regulatory processes, and immune defense. We said that proteins can further fold into more complex structures compared to peptides. Unlike peptides, proteins have the ability to fold into more complex structures guided by interactions between amino acid residues within the same primary structure. The primary structure of proteins consists of a long unbranched chain of more than 50 amino acids joined together by peptide bonds. This primary structure of proteins can further fold into secondary structure due to various interactions, including the hydrogen bonds that form between carboxylic group hydroxyl and the amino group hydrogen of another amino acid. The second interaction are the van der Waals forces that can act between nonpolar side chains. These interactions contribute to protein folding into secondary structures. The two common secondary structures that can form when the primary structure folds are alpha helix and beta sheet. Now moving to the protein tertiary structure. Protein's secondary structure can undergo additional folding to form fibrous or globular structures. Those structures are the tertiary structures of protein. The tertiary structures are also driven by various interactions between amino acid residues. The interactions that can result in the tertiary structure of protein include hydrogen bonds between carboxylic and alcohol side chains, and hydrophobic interactions, disulfide bridges, and van der Waals interactions. Also, ionic bonds between oppositely charged side chains have a role in interactions that result in the formation of the tertiary structure. The tertiary polypeptide chains interact similar to the secondary structures and fold into the quaternary structures. The individual polypeptide chains join together, guided by specific interactions, to create a protein complex quaternary structure. Now, if you look at this representation of the formation of the primary, secondary, tertiary, and the quaternary structures of protein, we can see that the amino acids bind together to form protein's primary structure, which is unbranched long chain. This primary structure folds into two possible secondary structures, alpha helix and beta sheet. As folding continues, the protein adapts its tertiary structure, globular or fibrous. Protein's polypeptide chains further assemble into complex quaternary structures. Now we have to remember one thing. The quaternary structure is formed of subunits. Protein subunits are polypeptide chains or single protein molecules that join together to form protein quaternary complex structure. This means subunits that come together to form the quaternary structure can be secondary or tertiary subunits.